So dear friends, warm greetings to you all. With hope that you are positive in spirit and testing negative, I, Dr. Sachin Kadam, Chief Technology Officer of Advanced Cells Group, welcomes you in today's webinar, which is on historical events happened in stem cell research and therapy so far. The history of stem cells and stem cell therapies present a unique story of inspiration. And also this story embodies the curiosity with which scientists work to solve the mysteries of the biological world, from the eras of basic science discoveries to modern days advanced translational medicine. By now, we all know that what stem cells are and why they are so popular or special. Even if we open daily newspaper, we come across a news which is related to stem cells. So it is important for us to know how these stem cells actually came in picture, who discovered them, and what is the history associated with the stem cells. So let's begin with the today's webinar. So stem cells is the most common name of these specialized cells we know. But there is a historical association, or the naming associated with these cells has a history behind it. Scientists named and renamed these special cells of body as and when they discovered something new about them. Thus, the evolution in stem cell nomenclature was a continuous process even today. MSCs or mesenchymal stem cells were originally named by Alexander Fredestin as a colony forming unit fibroblast, and then osteogenic stem cells after their discovery. However, since this time, a number of terms have been used to describe mesenchymal stem cells, including stromal cells, which was proposed in 1988 by Dr. Maureen Owen to indicate that the cells reside in stromal rather than hematopoietic compartment. Instead of highlighting the cell's compartmental origin, Dr. Arnold Kaplan proposed the term mesenchymal stem cells in 1991 to emphasize the self-renewal property and differentiation property of these cells. However, this nomenclature was changed by James Dennis, who suggested that the cells may be progenitor rather than stem cells. As a result, the term mesenchymal progenitor cell was proposed. In 2000, Paolo Bianco and Pamela Robey coined skeletal stem cells to specify that the cells give rise to component of the skeletal system. While just 2 years later, the term multipotent adult progenitor was proposed by a scientist, 𝐂𝐭𝐞𝐦 and his colleagues. To describe the multipotent nature and potential progenitor status of these cells. Since no direct evidence demonstrated the ability of mesenchymal stem cells to self-renew and differentiate in vivo, in 2006, the International Society for Cellular Therapy proposed the term multipotent mesenchymal stromal cells. More recently, in 2010, again, Arnold Kaplan suggested that acronyms MSCs stand for medicinal signaling cells, to reflect that the primary therapeutic benefits of MSCs may be attributed to secretion of bioactive molecule rather than tissue regeneration. However, mesenchymal stem cell is the most used term till date, and we are still using that. The existence of stem cells has been known for a surprisingly long time. Before and during the 1800s, scientists observed for the first time that the basic unit of the cell, basic unit of the life is called the cell, which is they have observed underneath the microscope on the discovery of microscope. Scientists often call themselves where these cells arose from. In mid 1800, Polish doctor named Robert Remarque first created the theory that all cells arose from pre-existing cells. Rudolf Virchow, a German doctor, often called as father of pathology, first accepted Remarque's work and built upon Theodor Schwann's cell theory, declaring omnis cellulae cellula, means all cells arise from pre-existing cells. The first major step in identification of stem cells came from Russian-American scientist Alexander Maximov. Maximov's Unitarian theory of hematopoiesis verified that all blood cells develop from single precursor cell. After Maximov's theory of stem cells origin were verified, stem cell research began to gain momentum. In 1961, biophysicist Dr. James Steele and hematologist Ernest McCock at the Ontario Cancer Institute were unaware about the journey on the path of discovering stem cells and the great potential these cells hold for treating many diseases from blindness to spinal cord disease and so on and so forth. During their study, what they did was they had a bunch of groups of mice. These mice were irradiated with enough X-rays to kill them within 30 days if they did not receive a transplantation of bone marrow cells. The mice were also injected with varying number of cells in order to the researchers to determine how many cells it would take to keep the animal alive. On one Sunday morning, several days after injecting these cells, McCock examined samples taken from the mice. This hematologist noticed small lumps on the spleen of mice, one lump for every 10,000 cells injected bone marrow cells. Dr. McCock's observation that the number of clumps which they thought as colonies was linearly related to the number of transplanted marrow cells suggested that colonies might be clones, and these clones they derived from a single cells. They published this accidental finding in the Journal of Radiation Research, proving the concentrate existence of stem cells, and here the race for innovation is actually started. At the same time, Dr. Louis 𝐶𝐚𝐭𝐤, a molecular biologist and Dr. Till's mentor for his postdoctoral work, was the lead investigator for the studies that revealed the cells that gave rise to colonies had among their descendants cells that could themselves give rise to new colonies. So colony forming cells could self-renew. So this is what the theory Dr. 𝐶𝐚𝐭𝐤 was proposed. 𝐶𝐚𝐭𝐤's work, also published in 1963, combined with many prestigious findings, convincing that Dr. Till, whatever had observed, is nothing but a blood-forming stem cells. Now coming to a major discovery, the technique of culturing mouse embryonic stem cells from inner cell mass of pre-implanted blastocytes. And this was done almost 40 years ago. In 1981, Evans and Kaufman isolated embryonic stem cells from early embryos, known as embryoblast, from mice, and established them in cell culture. These early embryonic cells have the potential to differentiate into any of the cells of the adult organism. They modify these stem cells genetically and place them in womb of female mice so that they would give birth to genetically modified offspring. This was also achieved by Dr. Gail Martin independently in the same year. Eventually, Evans was able to isolate the embryonic stem cells of the early mouse embryo and establish it in culture. Post-discovery of human embryonic stem cells, research related to stem cells saw many ups and downs, and even US federal government's strong interference. In 1988, United States federal funding of embryo research is approved. It is later vetoed by President George H.W. Bush. In 1994, President Bill Clinton repeals an order he initially placed on allowing embryonic research to occur. The next year, federal funding for embryonic research is banned. Widespread public interest in stem cell research soon ushered in new age of endeavors. In 2001, President George W. Bush prohibited the federal funding of human embryonic stem cell research for all stem cell lines derived after 9th August 2001. The policy enacted did not apply to state funding. In 1998, Dr. James Thomson, a world-renowned stem cell researcher, was the first scientist to isolate human embryonic stem cells. His creation of human embryonic stem cells from human embryo created much turbulence, both positive as well as in a negative way, in the community. The human embryonic stem cells opened the door of stem cell therapy, at the same time faces the criticism and created ethical turmoil due to sacrificing living human embryo. Later, Dr. James Thomson also contributed in discovery of induced pluripotent stem cells. The discovery of induced pluripotent stem cell was hailed as a revolution in stem cell biology and stem cell engineering, because many of the ethical concerns associated with the embryonic stem cell research could not be sidestepped. Why? Because these iPSCs were derived from fully differentiated adult cell. In this case, Yamanaka and Thomson, they have derived it from adult skin. Dr. Yamanaka and Dr. Thomson were awarded numerous awards for their work, which also include Nobel Prize. Most discoveries, for the matter scientific discovery, happened accidentally when we are not prepared for it. As a researcher, one needs to be more cautious and aware of each and every single step in the set of experiment. Multilineage differentiating stress enduring cells, also called as moose cells, came in picture in the same manner. It was the mistake created in a hurry to attend wine party by Dr. Devas. She happened to isolate these cells from mesenchymal stem cells. We can see moose cells in more detail in next upcoming webinar. For now, if we have to see the properties of these novel cells, these moose cells are positive for the pluripotent marker stage-specific embryonic antigen SSEA3. They are stress tolerance pluripotent stem cells. They are part of mesenchymal stem cells and fibroblasts, as well as they can isolate from human bone marrow mononucleated fraction. In 1990, Dr. Donald Thomas won Nobel Prize for his discovery in cell transplantation in the treatment of human diseases. Dr. Thomas is known for the first allogenic hematopoietic stem cell transplant. In 1981, Sir Martin Evans managed to cultivate what are referred to as embryonic stem cells from mice. This achievement opened the way to producing living mice with modified genome. This advancement allowed Mario Capici and Oliver Smith to breed live mice with specific gene inactivated, making it possible to elucidate these genes' function. This research was awarded Nobel Prize in 2007 and same was given to Sir Martin Evans. The 2012 Nobel Prize in Physiology for Medicine was awarded jointly to Sir John Gordon and Shinya Yamanaka for the discovery that mature cells can reprogram to become pluripotent, that is, induce pluripotent stem cells. Now coming to the stem cells in regenerative medicine, according to WHO report, today more than 50,000 transplants are carried out annually worldwide, and they are increasing each year. Stem cell transplantation also remains the last hope for patients with advanced diseases. Although stem cells existence was not known to us, its beneficial effect was well documented even in 500 BC, when in Ireland, a person with major trauma was treated with xenogenic bone marrow transplant. Unfortunately, the patient died on 7th day due to major trauma, but the first case was registered as the xenogenic bone marrow transplant in way back in 500 BC. After that, the first human bone marrow transfusion was given to a patient with aplastic anemia in 1939. This patient received daily blood transfusion and an attempt to raise her leukocytes and platelet counts was made using intravenous injection of bone marrow. Then after World War II and the use of atomic bomb, researchers tried to find ways to restore bone marrow function in aplasia caused by radiation exposure. The first allogenic hematopoietic stem cell transplant was pioneered by E. Donald Thomas and reported in the Journal of New England Journal of Medicine on September 12th, 1957. This study, 6 patients were treated with radiation and chemotherapy, and then they received intravenous infusion of marrow from normal donor. Only 2 patients managed to engrafted, and all died by 100 days post transplantation. At that time, little was known about histocompatibility antigen, and no one tried to match donors and recipient. Many tried, failed, abandoned the field, but Thomas believed in potential of this treatment. In the mid-late 1960s, somewhere in 1968, method to identify type HLA in human were developed, which allow for donor and recipient HLA matching. In 1969, Thomas initiated a clinical trial program in Seattle for allogenic hematopoietic stem cell transplant. In 1977, the Seattle group reported 100 transplantation with chemotherapy and radiation therapy in 54 patients with acute myeloid leukemia, AML, and in 46 patients with acute lymphoblastic leukemia, ALL. Of these 100 patients, only 13 patients were remain alive without disease for up to 5 years post hematopoietic stem cell transplant. However, this small cure rate only encouraged Thomas to try and apply allogenic hematopoietic stem cell earlier in the course of acute leukemia, and in 1979 he reported a cure rate of 50% in AML patient transplanted in first remission. Perhaps the most important thing Thomas found in his work was power of immune system to eradicate cancer. And remember, the Nobel Prize was given to Thomas for this particular study. 1973, in New York, the first successful transplant between 2 non-related people was performed. when a young boy received a stem cell transplant from non-related donor identified compatible through a Danish blood bank. In 1988, the first umbilical cord blood transplant was done effectively in Paris. When stem cells use in therapy establish, the need for stem cell donor arise. Okay, and to fulfill this need, in 1972, the International Bone Marrow Transplant Registry (IBMTR) was established for documenting hematopoietic stem cell transplant outcome data. By that time, transplantation were done in 12 centers performing about 50 odd procedure a year. In 1974, the European Group of Blood and Marrow Transplantation (EBMT) was established for European collaboration in the field of hematopoietic stem cell transplant. The first unrelated donor transplantation inspired in 1986 the foundation of the National Marrow Donor Program (NMDP), and in 1988 Bone Marrow Donor Worldwide was founded. This organization unifies more than 23 million registries in 73 odd countries and almost 6 lakhs cord blood unit from cord blood banks in 32 countries, which is very remarkable. After all these discoveries, if we really want to know where we stand today, we really have to see the stem cell publications or stem cell research going across the globe. If we have to see over the year development in stem cell research, we found out that with the number of publications happened so far, this number is growing every year. I just did a PubMed search for the term stem cell and found out that this growing number of publications have reached to around 350,000 publications in one single year of 2019. That shows what importance stem cells have gained in last half century. We know that with enormous growth in science and technology, medicine is also growing with high speed. For many incurable diseases, we are trying to find the medicine or a treatment. We have many keys in our hand that can use to open and close door of knowledge. But from the bunch, only one key can do the needful. Every lock has different key. Same way, one standard stem cell therapy cannot cure different individuals. The research is still on with stem cell application in regenerative medicine. US FDA have recently categorized stem cell as a drug, but stem cell therapy is still considered as an experimental therapy. One need to remember this. To understand more clearly where we stand today in terms of stem cell therapy, let's have a look at clinical trials happening around the globe for the stem cells application in different diseases. All around the globe, somewhere around 6,000 clinical trials have been registered for stem cell and stem cell therapy. Among all, US is leading the highest number of registered clinical trials. For many different diseases, right from neurological to cardiovascular to diabetes, etc., clinical trials are on using stem cells. Of these, around 50% of studies are in phase 2 or 3. Many clinical trials have completed their studies to confirm the potential of stem cells to treat otherwise non-treatable disease via conventional therapies. The rapid progress towards successful commercialization of stem cell therapies drives growth of mesenchymal stem cells as a major type of stem cells. So, when we consider a stem cells research or a stem cell therapy, one need to know that stem cell therapy grows in therapeutic value for its ability to protect cell repair damage damaged tissues or damaged cells and promote new cell growth. MSCs afford promise in tissue injury and immune disorders with somewhere around 23.1% of MSC-based clinical trials in 2018 focused on bone and cartilage. In the year 2018, Mesoblast Limited completed phase 3 clinical trials of MSC derived from bone marrow for the treatment of pediatric patients who have failed to respond to steroid treatment for acute graft versus host disease. Global opportunity in regenerative medicine drives interest in the tissue repair capabilities of stem cell therapies. Development in mesenchymal stem cell manufacturing platform, including bioreactors, drive MSCs derived products from discovery to manufacturing and commercialization is going on, and it's still under process. So with this, I would like to conclude my talk. If you have any questions, most welcome. If due to time constraint, if I'm not able to answer those questions here, I'll try and answer these questions in our Facebook page or social media handles. Thank you so much.